like to
Thank you.
move on to our next speaker, that is Mr. Chandrasekhar Bhupathi. He is working as a group head for clinical pharmacology programming group in India. He has close to 14 years of programming experience in clinical trials. He has been with Novartis for the past 12 years, and prior to Novartis, he was associated with Cytal Pune. He has completed his PhD titled Some Problems on Statistical Approaches in Clinical Trials in August 2020 from the Department of Statistics, Osmania University, Hyderabad. So, welcome, Mr. Chandrasekhar. His topic for today would be clinical pharmacology in oncology clinical trials. So, welcome again, and over to you.
Thank you, Parul. I'll share my slide. Please confirm. Please let me know if you can see the screen.
Yes, we can.
Thank you. Uh, First of all, thank you for giving this opportunity to present this topic. And as previous speakers already covered many of the topics related to oncology and the different designs and the burdens where we stand in research in global and in India, and also the role of programmer and role of statistician, and especially in oncology. So here today I would like to share uh, like the importance of clinical pharmacology in oncology. So I would like to go very high level overview of this clinical pharmacology in oncology. So with this, basically, maybe. So disclaimer again, standard disclaimer. I know these views I'm going to present in this topic are my personal and does not reflect Novartis views. In the agenda, we are going to, I'm going to just overview on what is clinical pharmacology and the importance of clinical pharmacology, and what are the study objectives of this clinical pharmacology studies, and how the study designs and what analysis we do in this clinical pharmacology studies, and what are the some common challenges we in the terms means a programmer and a statistician come across in this clinical pharmacology studies. So I mean, introduction basically, what is clinical pharmacology? Clinical pharmacology is nothing but it's like study of drugs in humans. So it's nothing but this is what we do clinical trials. The clinical trials nothing but test experimenting the uh, new tested the new drug in the humans. So here in the clinical pharmacology we study basically the drugs in humans. So basically when you want to study about this piece, we need to look in two ways. So like when the new drug is given to the patient, so we should look into is what is happening to the drug and what is happening to the patient. So we have two ways to look here. So the first one, what body does to the drug is called as a pharmacokinetics. This we basically understand how what administration and what is happening to AD, like absorption, distribution, metabolite and excretion, and through urine concentration, plasma concentration, we come to know these things. And next one is what drug to the body. So what body does to the drug is PK, and what drug does to the body is pharmacodynamics. This is basically nothing but the effect. So like how the dose is response, basic dose response thing, and mechanism of action, signaling pathway. So we study in PD basically about these aspects. So in simple, I put in a graph basically. So in pharmacokinetics, we understand concentration versus time. In pharmacodynamics, we understand effect versus concentration. And coming to the next slide, basically, and the importance. So I'm not sure, but did any one of you ever open whenever we purchased a medicine package or a complete box of pack? We generally have a leaflet inside it. If you ever get a chance to look into it, you might have familiar with the one I'm going to talk. But if not, next time when you get a chance, please pay just a couple of seconds attention. It generally explains about full prescription, about the information like what indications and all these things, dosage and information, it covers many of these things. And when you notice basically in a leaflet, this leaflet, all these, the one which highlighted in blue color, are basically coming from a, most of these things are coming from a clinical pharmacology studies. So I'm just going to, and especially there is a dedicated section that highlights the clinical pharmacology section. So I just want to highlight the importance with this small leaflet. But next time when you get a chance, please spend some time. So then we'll go to like what are the endpoints. Now we understand what is PKPD and what is the importance of the clin pharm trials, and what are the endpoints that we use while analyzing this clin pharm studies, the PKPD studies. So PD, as I mentioned, basically it's an effect, that means outcome of measure basically. So, and it could be like quantitative measure or a binary measure, basically whether an event occurred or not, or a time to occurrence of an event. All these things will be part of PD endpoints. These are few, it's not limited basically. I just highlighted a few things. And coming to PK endpoints, basically as I mentioned, the concentration over time basically. We draw the curve and we try to get some pharmacokinetic parameters we call AUC, area under curve, and the maximum concentration, and the what's Tmax, what we call time at the maximum concentration. So there are few, there are many things. I just highlighted very few. So these are the endpoints which we use, which we refer while analyzing the PKPD trials. So now we'll go to like study objectives. Like what are these clinical pharmacology study objectives? And again, as I mentioned, these objectives may not limited to these things. So here let us say, I just mentioned like ADME studies and accumulation studies, bioavailability, bioequivalence, food effect, drug-drug interaction, all these things, dose proportionality, hepatic impairment, renal impairment, ethnic sensitivity, thorough QT study, and obviously these are not limited, many more are there. And here we'll spend some time to understand. I don't want to just put the names. I want to really explain basically how these things. So ADME study, we know basically it's FDA recommended study. It's mostly it's obviously the phase 1 study, and which helps to understand the absorption, distribution, metabolism, and excretion of the drug. And when it comes to the bioavailability, the objective here is to, how can we say the bioavailability? This means basically the rate and extent which the active drug substance or therapeutic moiety is observed from a pharmaceutical, observed from a pharmaceutical and becomes available at the site of action. That means we can say that when we say two drugs, basically they have to, like, the substance in general should have the availability should be same, basically the rate and extent which the substance or its therapeutic moiety is delivered from its pharmaceutical should be, in general, it should be same. So when it comes to the bioequivalence, we say that we should say that these both two are equal in biologically, two drugs basically. I would say um, the absence of significant difference between the rate and extent of the drugs, basically I would say that the test and the reference drug should be um, same, you know in terms of there it should be similar conditions basically. So it's again, this is also a recommended study, and obviously we have ICH guidelines, clearly provided guidelines on a bioequivalence of requirements, and we have basically a rule that it should be satisfy the ratio of the test and reference drug should satisfy a 0.8 to 1.25 range when we analyze the results. So, and obviously the food effect, the objective is to assess the impact of food with the drug on the drug base on the body basically. That means we assess here a drug with food and without food, or with low fat food or with high fat food. So like that, uh, this food effect study will be conducted. And the impairment studies, either hepatic or renal impairment studies, basically it is basically to determine the dosage recommendations for this impaired patients, renal impaired patient, renal or hepatic impaired patient. We need, and this is also recommended by the FDA. These studies are so. These are few. Again, as I mentioned, I'm not going all the studies. I just want to give importance of few things. And now coming to the next one. for study designs basically. So ADME, it's mostly I just want to highlight in this slide the varieties of study designs we use for PKPD trials. So for ADME, we typically prefers an open label and it should be mostly a single dose study. And for organ impairment studies, single dose basically if drug metabolites exhibit a linear and time independent PK. Otherwise, recommendation will be multiple dose if drug and metabolites exhibit non-linear basically and time dependent PK. So BABD, food, DDI, mostly we crossover design. I mean, there may be a parallel designs, but mostly it's go via crossover designs. And TQT basically the complexity and thorough QT studies. Basically, we take a time matched baseline and average of triplicates. Like ECG measurements will be collected multiple times for a same visit, basically with a 5-minute interval or a 3-minute interval, depending on the study. But they all consider the same visit basically, and that's called a triplicate. And for a baseline and post baseline, we have to define a complex rules stating that we have to take average of these records as a baseline or minimum or maximum as baseline depending on the study and other requirements. And statistical models, what statistical models we use basically. So these parameters, especially coming to the PK parameters, these will be log transformed before any analysis purpose, and that means before to execute any statistical model, these will be log transformed, and then we run a statistical model and then back transform once we have statistical model results. So mostly, here also I would like to highlight the difference, like what kind of variety of statistical models we use for analyzing our PK or inform trials. ADMA studies, mostly I'm sure few people who are experienced with ADMA trials hardly will have a single digit number of patients, like hardly 6 or 8, mostly 6, my experience basically. And with the 6, the study will be conducted, and we'll have obviously many reports, and what we produce in these reports is that mostly summary statistics, and obviously listings will be there. So a simple summary statistics in ADME. When it comes to a DDA study, a drug interaction study, basically we may use a linear mixed model considering treatment period and sequence as fixed effects and subject within sequence as a random effect. Example basically this again. And for a bioequivalence study or a renal impairment study, we can use analysis variance or analysis covariance depending on the covariates mentioned in our study designs. And sometimes when you say RP2 RP recommended phase 2 dose studies, we use Bayesian logistic regression model. All these things we use. And apart of these things, basically, yeah, that's it. And contributions, clinical pharmacology trials like PKPD trials contributions. Obviously, for each study we have a CSRs, and we have to write the clinical summary reports. And these studies, these clinical trials also part of POPIC analysis, population PIC analysis, and also use exposure response analysis, example to to determine a dose regimen. And Hanumanta covered about SCE and SES, summary of clinical safety and summary of clinical efficacy. So along with that, for clinical pharmacology, we have a dedicated section called SCP, summary of clinical pharmacology. So again, this is like pooling of multiple, as Harmanta explained. When we are doing a submission at a compound level, we need to pool multiple clinical trials and explain the clinical pharmacology properties in that SCP. Obviously, we do support different packages. All these clinical trials, when it's going for submission, will be part of different packages. As part of package, we do all these things. And once submission is done, we do receive a lot of HAQs. So this work, the point I want to highlight with this slide is that the work, what we are going to do here is very much important and plays significant role in individual CSRs and also in submission and also in decision making. And a lot of exploratory work also happens around these clinical pharmacology trials. So, yeah, what are the common challenges we face. We means as a programmers or statisticians together, we face basically during these clinical pharmacology trials. So as we just now had from Vincent stating that the new designs and all these things we need, that's one of the need. We are getting this kind of new trials like multiple objectives with one trial, like which quite common example I put food effect combined with the DDA study, drug interaction. For statisticians, they need to place the multiplicity adjustment, power adjustment, and also sample size, all these things. And for a programmers, basically, we have to define specifications in such a way that address our specifications, programming specifications, satisfies for both both the designs or both the objectives. So obviously, it seems one study, but actually they combined two studies into one protocol. So obviously the workload will increase, the resource will increase, the number of DFLs will increase, more challenges, operational challenges basically. And other one is when the most common thing is that for coming to the PKPD, the sample collection, PK sample, blood collections or blood draws, when it is unscheduled samples, protocol mostly, almost all the studies, the protocol defines clearly what to do with the scheduled sampling. This is mainly, please, this is mainly for the PK perspective, these lines basically. The unscheduled samples, I'm speaking in terms of the PK samples. So protocol has a clarity on how to handle the scheduled samples, how to map these scheduled samples to which dose and all these things. But unschedules, generally, the clarity will not be there, and we will definitely struggle if you want to map these unscheduled samples to relevant doses. And combining this concentration data with dosing information will also another challenge. And obviously inconsistency of analyte metabolite naming convention or missing information, especially since these data PKP data coming from a laboratory information processed by different labs mostly. And when we receive data for programmers, we always face challenges. Protocol says a standard name called ABC, but however we receive it as a ABC1 or ABC2, sometimes a different name. So this is quite challenge to match this thing, because we are programmers. Sometimes we may not have a scientific knowledge to understand, oh, this name belongs to what is protocol mentioned name, or a simple thing. Sometimes a lowercase, upcase, lowercase issue, lowercase letters, uppercase letters, and with additional characters, these things. And because of the protocol design, one sample may need to map to multiple doses. Classic example and standard example, you say that a sample been collected after 10 days, that should be considered as a post dose for a previous dose versus a pre dose for the current dose. That means one sample only, but we have to map it to two different doses, which is a common challenge. And another problem is that because of this data, mostly it comes from the external labs and the third party data most of the times basically, or even if it is internally also, there will be delay in availability of this data, because the PKPD data basically we mostly receive the safety data first, then we receive this data. So obviously there will be delay when the data is coming late. The more pressure will be the programmer to create deliveries for this data. And also PD endpoints, especially the PD is a big another challenge, because PD is a general, it's like effect, and this PD can be anything. It can be part of our lab, it can be part of our exploratory data, sorry, it can be part of our PCG, it can be part of our PK data itself. So it can be sometimes it can be a separate data also. So when we're coming at a part of a new data set, we'll have obviously more challenges to handle this data. And also, as I mentioned, data inconsistencies. Even we receive, but sometimes units in SAP versus what we received, definitely it will change, and we don't have a conversion rule. Okay, how to convert what is the right thing. Obviously Google is there to help us, but we should not rely only on Google, right? We, as our previous speakers mentioned, we are in a highly regulated environment. We should have a proper documentation of what we are doing and how we should do. So these are a few challenges. I'm sure there are many more. These are few challenges. Yeah, that's it from my side. Yeah, if time is there, I'm happy to take few questions.
Thank you, Chandru. It was for for this session. There are a few questions I can see. Let me pull it up. Yeah, so this is about missing data points. How how can we calculate the missing time points in urine analysis? Have you come across any maybe if you can elaborate a little bit on handling missing data?
Missing data itself, it's a huge concept, as we all know that. And obviously the protocol, first thing, if this question asked by a programmer, we should, as part of our checks while we review protocol or SAP, these are the first things we're supposed to check and ensure that what we should do if there is a missing data. It could be a result, it could be dates. If there are missing dates, how do we handle? Example, classic example, adverse events. We should know basically how to handle if there are missing dates, and if these dates are very important to us. Same thing, if there's a result is missing, and this particular endpoint result is important to us, we should always clarify at the very beginning while reviewing our SAP and protocol stage only. There are many methods. It could be like, sometimes people take last observation carry forward. Sometimes they take average, especially in ECG. Sometimes, as I mentioned, triplicate case, when we're taking average instead of triplicates average, RAP or the SAP may suggest to take available replicates average. Sometimes if you have only two replicates, you will take average of only two instead of three. So this kind of clarity should be there in protocol, and that should also have clearly mentioned the SAP. There is no one straight answer basically here. It all protocol driven, and SAP has to cover these things.
Thank you, Chandra.